So some of my Monday classes are also canceled because Tuesday's canceled, but I will be there in office hours, same time, same place on Monday. Okay. So,
There's no class on Tuesday?
there's no class, not tomorrow, next Tuesday is Veterans Day. Go hug a veteran, thank them for their service.
It's
nope,
Tuesday.
Tuesday is the day that's off.
You come Monday, but you can't come
Monday you have
Tuesday.
classes, we will have classes next Monday and here. Tuesday is a no class day.
4-day weekend.
Yeah.
All right, any other course logistic questions? I had a question up front about the Connect, the online system Connect that you guys, those of you with the 5th edition textbook, there's a few chapters that I didn't quite pull into that, so I'm not caught up on that. I will get that cut up for you guys ASAP. Okay. Other stuff? All right, so in terms of describing types of mutations in DNA, so that's kind of where we've been talking about. We have one more mutational classification or characterization to go over, and then we're going to get into repair pathways. Okay, so after we discuss repeat expansions, the whole rest of what you guys need to know before the second exam is all about how do we repair these mutations. So how does a cell evolve to have different mechanisms in order to fix these things, okay? I really do anticipate that we're gonna finish with time on Monday to go over some practice problems that I put together. So I think I'll treat this the same way I did the Monday before you guys' first exam, where we spent some time finishing up any last lecture material, and then I'll create some practice problems for you guys that we can go over in class. So Monday's gonna be more about reviewing and practice problems. I have your homework back too. Again, you should be able to see that up on iLearn, your grade for that. I'll finish up early tonight and let you guys know. So just please be respectful and stay quiet and settle down at the very end of lectures so that I can get the homework laid out and have you guys be able to get a chance to come pick that up if you so desire. Okay, so this whole repeat expansions bit. So most of the rest of what we talked about, we're really involving, one nucleotide change, either chemical changes to the structure, or you're swapping the nucleotide out, you're deleting it, or inserting it. This topic of repeat expansions this is large-scale mutations. They involve either deletions or multiplications of a repeat unit. So often this is in the form of 3 bases. So you can see repeats where you'll have a 3-base repeat that's then that same 3 bases is repeated over and over and over and over again. So they often refer to this as TNRE, this phenomenon. Okay? And it stands for trinucleotide repeat expansion. Okay? So it's referring to having that 3-base repeat expanded beyond what's considered normal, or what we see in a wild-type normal individual. Okay. So it's an expansion of a 3-base repeat. Again, these are spontaneous, so this isn't, this actually falls, because we kind of, I guess, ended up talking about more external causes that can induce mutations. This is more of a spontaneous type of mutation. So this is something that happens in our cells, and I'll briefly kind of talk about the mechanism of that. For us to discuss this trinucleotide repeat expansion topic, I'm putting it in the context of a case study. So there's a particular human disease, and it's called Fragile X. So this is in humans, and it involves a trinucleotide repeat expansion, so that's where the disease comes from. The reason it's called Fragile X is cytogeneticists can look at chromosomes under the microscope, and they can see the abnormal X chromosome. So, normally your X chromosome, you have centromere, and you can see sort of a imagination where the centromere is, the chromatin kind of dips inwards because it's tightly wound at that point. On a fragile X, the chromosome, we can kind of see it as, under the microscope, there's an extra, and it's often kind of still pinched inwards, but there's sort of an extra repeat. So this is where the expansion is. And it happens to be towards the end of the chromosome, but this trinucleotide repeat is way, way, way longer than it normally is, and it's also tightly wound down, and we're going to talk about that. There's a lot of silencing that happens at that region. So cytogeneticists can look in the microscope and see it as this strange kind of more narrow region at the bottom of the chromosome. And it tends to be prone to breakage and other things. So this is kind of where the name Fragile X comes from. Okay. It's inherited in a dominant, and then of course X-linked fashion. And it's a, sorry.
Does that have a centromere?
Yeah. Sorry, I didn't make it dark. I know. It's common. Yeah. It does have a normal centromere. It just has an extended Q arm, the very bottom arm, and it's kind of pinched. This is actually one of the most common inherited forms of mental disability is Fragile X. So the disease is characterized by abnormal cognitive development and things like that. I think the last numbers I checked, they kind of change quite frequently, but there's about 1 in 4,000 males that are affected with this, and about 1 in 8,000 females. And that number is increased actually over the years since I checked it before. So quite fairly common actually in the human population. And it results from one of these 3 base pair repeats. It's a CGG repeat, so cytosine, guanine, guanine, and then that's repeated many, many times over and over again. So this disease is a result of changes in the number of times that this 3 base pair segment is repeated over and over again. And the particular region where this repeat is, it is near a gene. It's not actually part of a gene. It's in the untranslated region, which we haven't gotten into a lot of detail, but you guys should remember from your previous classes thinking about UTRs, 5' and 3' UTR, untranslated region. Usually those are regions that are flanking on either side of the gene, right? And we'll talk more about untranslated regions as we get into regulation of genes and eukaryotes in particular. So it's in the untranslated region, FMR1 gene. So this gene is named, it's Fragile X mental retardation 1. Most folks now refer to it as FMR1 because, of course, as a society, we sort of moved away from using the term retardation, and we describe these kinds of diseases more on a cognitive disability sort of level. But that's, you guys are familiar with those changes with what's socially acceptable and nomenclature at the time. And when this gene was first discovered, this was long before we established a less derogatory or negative term for these kinds of disorders. So it's called FMR1 gene. This gene, so the protein product of this gene, what it does, so this protein, or the product of this gene, okay, is a regulatory protein. And in particular, it regulates, for your purposes, it regulates translation of many other proteins. And it's also a crucial component, the product of FMR1 is crucial for development of synapses. So what does that tell you, the fact that it's involved with making those synapses?
The neuron connections.
Yeah, all of your neural development. If you don't have proper signaling happening between two nerve cells or all your nerve cells, you guys can kind of see on the biological side of things how this would really affect cognitive development and function. Okay, so if you knock out a gene or affect the ability of a gene to produce a normal product, that one is involved in translation of many other proteins and two is involved in neurobiological development, this creates a lot of problems when we disrupt it. So what this looks like, because there's a certain number of these repeats that are normal, it's sort of these kinds of diseases, this trinucleotide repeat expansion stuff is pretty strange because there's it tends to be, Huntington's disease is another example of this. There's a bunch you guys can find. There's a set sort of range of repeats that's okay, that we don't see any phenotypic abnormalities. And then all of a sudden it goes way beyond and we see, even up to hundreds of repeats longer than what's considered normal, and we'll start to see a disease phenotype. So it's a weird, and especially in particular when you're talking about repeats that are more in a regulatory region. So the untranslated region of FMR1 isn't directly affecting the amino acid product, right? Because it's not actually translated, but it's affecting regulation of this gene, the on-off kind of switch of this gene. Okay, so you can think about this in terms of number of CGG repeats. So we're going to kind of make a table, create a table here that gives you a sense of what's normal, what's considered a carrier for this disease, and then where we actually see the phenotype expressed for the. So, and I'll describe what each of these categories means, but what we have is number of CGG repeats, a phenotype, transmission. We're going to talk about methylation, which happens to play a role in this Fragile X syndrome. And then this last TRXN is my shorthand for transcription. Okay, so whether or not we actually see successful transcription of FMR1. So a normal scenario is one where you have, and these numbers vary a little bit, there's a little bit of flexibility, but between 6 and 60 repeats is a normal amount of repeats of these CGG trinucleotide. So our phenotype is normal. The transmission's stable. So what I mean by this, so when we talk about transmission, this is you can kind of think about it like the propensity or the chance, the likelihood that transmission from parent to progeny, that we're going to see another expansion. So the normal 6 to 60, we see that maintained faithfully. So if you had 60 repeats and you have a child, your child's going to have exactly the 60 repeats. Okay? So it's in terms of how stable it is, whether or not we're going to see an expansion on a vertical transmission line in terms of inheritance. Okay? There's no methylation in this area on a 6 to 60 repeat unit. And we do see transcription, so it's a positive for transcription of FMR1. Then there's this range where you go from greater than 60, but sort of under 200. And they often call this a pre-mutation, which is a new term for us that we haven't really discussed. Again, because these nucleotide repeat expansions, it's not like a hard line. Like if you have 199, you're absolutely going to have this disease, or 201. There is a range here. So they refer to this greater than normal repeats, but not so far that you have the full-blown disease as a pre-mutation. So 60 to 200 of these CGG repeats. And most of the literature will say that it's individuals with these types of numbers of CGG repeats in their untranslated region of FMR1 are mostly normal in terms of their phenotype. You may see some, they have recorded instances of some folks, some individuals who have some low-level neural phenotypes, so anxiety, some levels of autistic-like behaviors, things like that, okay? So, and again, with these, especially with the cognitive diseases, they are quite difficult to categorize phenotypically, as you guys can imagine. How do you tell whether someone's in a normal range of cognitive development? I mean, we can clearly point out when it's way off range, but again, like all the autism spectrum disorders and things like that, it can be difficult. So that's why I put this mostly in parentheses. Okay. The transmission of this, though, is unstable. Okay. So what that means is it's really prone to expanding further. When you have greater than 60 of the CGG repeats, it's likely to expand when you transmit it from parent to progeny. There's still no methylation, and transcription's still occurring okay. So that's why we see mostly a normal phenotype, a normal person, because of FMR1 is being transcribed and translated correctly. Okay, so when we get the full mutation, is when you have the greater than 200. So this person's affected, they're considered to have Fragile X. Their transmission is definitely unstable at this point, so again, prone to expansion. And then all of a sudden we see methylation. So what I'm saying by the CH3 is that region, that gene, all of a sudden when you have greater than 200 repeats, the cell methylates FMR1, okay, which eliminates transcription. You silence that region. The cell reacts to this great expansion of repeats by adding a bunch of methyl groups. So it's almost a form of epigenetic, not necessarily inheritance, but epigenetic gene regulation. Okay, so all of a sudden a bunch of methyl groups get added to FMR1. And by greater than 200, there's folks, you know, I've seen numbers of 1,300, it's 1,000. You can have up over 1,000 repeats. So huge. This region gets really expanded. And then that's when we see the phenotype expressed fully. And the reason I don't want you guys, for Bio 311, you don't have to really focus on the heavy detail on how these trinucleotide repeat expansions happen. But as you can imagine, if you have a CGG repeated over and over again, what conceptually I want you guys to understand that during replication, what happens is the DNA tends to make a hairpin loop. Are you guys familiar? We'll get into this again when we get into regulation. But because you have a bunch of C's and G's packed into an area that can hydrogen bond to each other, the DNA can kind of loop around like that. And what happens is DNA polymerase gets confused, it hits the hairpin loop, it slips off the DNA, it loses count, and all of a sudden you'll have huge regions of the DNA that are expanded on these repeat regions. So DNA polymerase in heavily repeated areas has a hard time faithfully replicating and copying exactly 20 repeats. It might end up with 21, 22, and so it starts to, those regions can start to expand. Okay, so that's what I mean by spontaneous. This just happens because of DNA polymerase's error prone in repeated regions like that. You guys are also not responsible for understanding the mechanism behind why it becomes methylated. This is just inherent with this disease. Heavily extended, untranslated region ends up causing methylation of FMR1. but I wanted you to see this as an example of how trinucleotide repeat expansion can result in the disease characteristics and phenotype.
Yeah.
And then the transcription category is transcription of the FMR1 gene.
Correct.
Yeah.
So both those last two columns in your table are referring to what's happening to FMR1 gene. Any questions about, so we just did this as an example of one of, there's many trinucleotide repeat expansions. Yeah.
Why would it happen more often in guys for order statistics instead of like there's more common in guys than women?
What do you think? What are your thoughts?
The 1X.
Yeah. Yeah. If it's inherited in a dominant fashion, you would expect to see, heavily male and female, but we see it a tiny bit more often in males just because they only have the one X. So females, sometimes we can see compensation by a normal FMR1 repeat on the other chromosome. Yeah.
And spontaneous, is it?
Yeah.
Sorry, I just thought of something. FMR1, that gene is making it so your synapses and all those proteins are good, right? It's not, and it's being silenced, and that's why you're affected.
Yes. Yeah.
So the name had, so FMR, the acronym, what is it stand for again? It sounded like that would be negative.
Fragile X mental retardation.
Yeah.
Is what the FMR1. So usually a lot of times they name
Yeah.
genes after the mutant form.
Okay.
Is that why you're?
Yeah,
does
so
that make
I
sense?
kind of looked at.
Yeah. So this is when this is functional, we want this functional, that's a good thing, and it creates all the normal development of neurons. Yeah, so a lot of genes, and that's not always true, certainly in the field of genetics, and as you guys are well aware, all the branches of biology, people are not consistent with their naming mechanisms. They name things for all different kinds of reasons, right? But in genetics, a lot of times genes are named after what's happening in a mutant or when it goes wrong. Okay, not always, but yeah, it's not a consistent. In this case, yes, it was named after the disease that was discovered, prompted the discovery of it. Yeah. Other? Okay. So let's move on to repair pathways. So we're still in chapter 16, 18, depending on what condition you have.
Before you move on.
Yeah.
Okay, she said that, she was asking if it was being silenced. Was it being silenced or was it being over-processed and
It's being silenced, yeah. So when you have this repeat region, this is in your untranslated, so all of a sudden you have a whole bunch of these CGGs getting bigger and bigger and bigger before or after the FMR1 gene. So not really affecting the actual gene sequence, but when that happens, the gene ends up getting heavily methylated, so that all the chromatin gets dropped down, it gets silenced. Yeah. It's a weird, not a direct result to the gene, but it results in expression changes in the gene.
Does it be like a form of imprinting then, since you have no methylation?
It would be a form of epigenetic, so the way you would actually talk about it would be epigenetic gene regulation, or epigenetic gene expression. But in terms of imprinting, we use that term when we're talking about genes that are normally regulated by methylation, and this one's normally not, that's an abnormal. And we also more refer to it as imprints or methyl groups that are passed from parent to progeny. And in this case, the expansion is the number of nucleotides, how many times it's repeated. That's inherited from parent to progeny, but the methylation is sort of a separate thing, yeah. But it's close if that's what, it brings to mind imprinting. That's good, because that means you're thinking of methyl groups affecting gene expression and all that, yeah. So it is definitely related in that sense. All right, so how do we fix this stuff? All right, so we talked about all these, we kind of had a negative discussion there, right? We're talking about all the bad things that can happen in your cells, and do happen all the time. Everyone in the room, we all have these mutations occurring, but luckily we have repair pathways. Okay, so there's ways that the cell has evolved to fix all the types of mutations that we've talked about. And a lot of these pathways, so don't get stuck in your head thinking about this pathway fixes this type of mutation, and then this pathway fixes this type of mutation. There are certainly pathways that work better for different types of mutations, but they're redundant. Okay, we have several pathways that can be operating at any given time in our cell, and I'll try to pull out the ones that are specific to certain cell cycle points, or that are really better for a certain type of mutation. But remember in your head, I mean, why would you want redundant pathways for repairing mutations in terms of natural selection and evolution?
In case one of the pathways mutates.
Yeah, I mean, right, if you only had one possible repair pathway, you're pretty screwed if anything goes wrong with that pathway, if you mutate a protein that's involved in that pathway, et cetera. So of course the cells, and DNA is such an important component for us for life. We've evolved, our cells have evolved to have many pathways and ways in which to fix our DNA. Okay. Mostly the bacterial ones are the best understood. So for the most part, I'll be talking about prokaryotic stuff. I'll point out when I'm referring directly to eukaryotic. And there's points in your books where it describes both. So again, I'll try to be really clear and just ask me if you're not sure if I'm talking about bacterium or eukaryotes, okay? Because these repair pathways are of course found in all of them. We just tend to understand bacteria better because it's a simpler system, so more research has been done on it. So your first one, and this one's definitely in, the way I'm gonna describe it is how we know it for E. coli. So bacterium is called BAIR. So that stands for base excision repair. So this particular pathway works on several different types of lesions or mutations. So this can repair oxidative damage, it's not very often for UV photoproducts, but it can be used for this. And definitely one of the major ones is whenever there's a uracil placed in DNA. So when uracil is stuck in as opposed to thymine, where it's opposite adenine, there is the common repair pathway for that. So this is base excision repair. So, try to put this out in steps that you guys can kind of think about. So this requires a lot of these, most of the pathways we talk about require enzymes. Okay, we have to cut our DNA, move things around. This is definitely enzymatic type of processes. So in this case, there's an enzyme that operates in there, and it's called N-glycosylase. So N-glycosylase is the one that actually recognizes the incorrect base or the damage or whatever is going on. Yeah.
Do we have to know that it's DNA and glycosylase, or just
N-glycosylase is fine. Okay, so this is the guy that comes in and recognizes your damaged base or whatever's abnormal. So if it's a uracil, for example. And it's going to cleave. Where do you think it cleaves? What bond? Based on the name. Between what and what?
The sugar.
Yeah, so between the sugar and your nitrogenous base. Perfect, yeah. So this is going to cleave that, the bond that's going on directly between base and sugar. So if you have uracil, for example, this enzyme is gonna come in and actually cut out the off your cell base. Okay? It's gonna leave behind a gap, essentially. And some of these mechanisms I'll try to talk more about when this is exactly happening. For basic excision repair, you can these enzymes are available, they're there in the cell, and they're scanning the DNA. So there's different points in cell cycle that our DNA gets, or bacterial DNA gets scanned more often. But in this case, the N-glycosylase will actually be scanning the DNA, and if it comes across something abnormal that it can recognize, it'll cleave off that base. And so at this point, you have a gap. And there's an endonuclease that comes in. And you guys aren't responsible for knowing the exact endonuclease, but it's going to recognize that gap. Or the missing base, essentially. And it's going to make an incision on the DNA, so it makes a nick. Nicks the backbone, and it nicks it specifically on the 5' side of the gap. So if you imagine an enzyme coming in, it's going to make a cut right there. And so again, remember, this is the difference between an exonuclease and an endonuclease. Endonuclease can come in and cut, like scissors, come into the middle of a DNA strand. It doesn't need an opening to come in, whereas exonuclease has to chew from an external end. Endonuclease can come in and make a cut in the middle of the backbone. So that's what it's doing here.
So look, on both sides.
Only on the one side, yeah. So just on the side of where the missing base is, because this presumably here, I don't know if this makes it easier for you to think about. I know I'm kind of drawing generic lines for the other base pairs, but you have an A and you have a U, and the U is the incorrect base stuck in there. So then you still have an A hanging out there, but you have kind of a blank on the opposite side of the A. Yeah. So then we recruit in DNA Pol 1. So polymerase 1 comes in. So again, I'm talking about E. coli here, so prokaryotes in this case. So polymerase 1 has what kind of activity besides polymerizing? What else can it do? Like what can it do with a nick right here? They have an endonuclease, a 5' to 3' endonuclease activity. They also have, a lot of them have a 3' to 5' and a 5' to 3' exonuclease, right? This has 5' to 3' exonuclease activity. So different from endo, which can cut DNA in the middle of the strand, exo, once you have this chunk missing, that's like a bare end. And, okay, it can like chop along that bare end. It can use its exonuclease to remove off bases. Okay. So this is DNA Pol 1 comes in and removes bases, all right, and can then fill it back in with DNA. So this is some of the proofreading repair mechanisms we talk about with the different polymerases. You guys are used to thinking, I think, more about polymerase 3, which is our major replicative polymerase, but you still have Pol 1, which can do things like excise bases and then backtrack and fill it in. Okay? So it removes the bases and also fills it in. And you guys don't need to know, the number of bases that are filled in or anything like that, but essentially it's going to remove a segment of DNA and then fill it back in using the other strand as a template, filling the complementary bases. So it doesn't just, this whole bare process doesn't just remove one base. It, you know, it may be that as Paul 1 goes along, it might remove a couple of bases and then fill it back in.
What calls it the damaged region? The damaged region?
Yeah, it's essentially doing it to the damaged region, but even if you have one base that's off, like a Uracil, it's still gonna take several bases off and then fill it. Yeah,
Why is it too that? Why is it too off?
The best, like, quick answer for that, to me, is the footprint that polymerase takes up is big. Like, it's such a big complex of proteins. It's not like it can just zoom in and like have the delicacy to like pull out one. I don't know why I always use one. Pull out one base and stick another in. It's like takes up so much space on the DNA. It has to like chug along and then back up and yeah. Okay, and we also need ligase at the very end. So it uses some of the basic enzymes you guys are familiar with. The endonucleases we've talked a little bit about in terms of restriction enzymes. Those are endonucleases, right? They make cuts, nicks on the DNA. And then we've discussed polymerases. You guys are real familiar with ligase. This N-glycosylase is maybe one of the newer proteins to think about.